3, 2, 1.
My name is JZundell, and today I'll be talking to you about the relative expression levels of SHP1, P53, and DHFR in various cell lines, and their implications within tumorigenesis. This is brought to you by the St. Leo Math and Science Department, and my mentor is Dr. Ian Duffy, and I have submitted all of my IRB forms. To begin with a little bit of background, SHP1 is a very important regulatory protein within T cells, B cells, and macrophages, very important in regulating immunity and dealing with pathogens within your body and effectively eradicating them. So SHP1 regulates various pathways controlling cellular proliferation, growth, and survival within these cells, allowing them to complete their processes within immunity. Next slide. DHFR is also another very important protein in immunity that is within and expressed within macrophages, B cells, and T cells. It's involved within the S phase of DNA synthesis, within cell cycling, within macrophages, before they've differentiated also B cells and T cells. So again, implicated in DNA synthesis within tumors, and a very important protein for regulating B cells, T cells, and macrophages. Next slide. P53 is another important protein I'm looking to analyze. That's involved with responding to DNA damage in response to various factors such as environmental stress, chemotherapy, and other various factors that damage DNA, like treating cancers and things like that with chemotherapy. So what P53 will do is it'll bind to DNA and mediate cell cycle control and often inhibit angiogenesis. P53 is mutated within about 50 to 60% of all cancers, so that kind of tells you its function within cancer and its conservatory function within evolution. So P53, as I mentioned, inhibits angiogenesis. Angiogenesis is a method employed by tumors where they direct blood vessel growth towards the tumor, essentially supplying it with nutrients for continued growth. And P53, being mutated, obviously isn't going to effectively deal with tumors and inhibiting angiogenesis. Next slide, please. So overall, as I had mentioned, SHP1, P53, and DHFR implicated within macrophages, phagocytic cells, T cells, and B cells in mediating and regulating cell survival, cell growth, and proliferation within immunology. So the purpose of this is to enhance oncological research by providing a means of expression of SHP1, P53, and DHFR within tumor-bearing cell lines versus non-tumor-bearing cell lines, and show their implications within T cells, B cells, and macrophages. Again, very important cells within immunity. Next slide. And I plan on doing this by utilizing cell culturing techniques within January, harvesting and splitting the cells in February, producing a protein lysate from the cells, and using that lysate within Western blotting in March, and also possibly ELISA and Western blotting utilize antibodies to bind a specific protein in question to detect their presence. And lastly, the last method I plan on utilizing is real-time PCR, which will help me analyze the genetic expression between tumor-bearing cells and non-tumor-bearing cells of the proteins in question by analyzing RNA. So much of the budget is already available. I do not exceed the budget because of donations provided by Dr. Ian Duffy and Dr. Tamar Gansa at the USF College of Medicine. This includes donations of tumor-bearing cell lines as well as non-tumor-bearing cell lines, including macrophage cell lines, T cells, and B cell lines. Also primers and antibodies for binding a SHP1 DHFR. DHFR and P53 antibodies are already present and have been used within biochemistry labs here. So through contacts, I will be able to acquire all of my materials needed for this project. The expected results include, it's difficult in vivo to conclude obviously in the body, but in an experiment in the lab, it's much easier to analyze protein expression. So SHP1, I'm expecting in tumor cells versus non-tumor cells, downregulation due to its implications within being knocked out within tumor cells. DHFR, I'm expecting overexpression due to its implications within S-phase cell cycling. The tumors are overproliferative cells, so I'm expecting DHFR to be overexpressed within tumor cell lines versus non-tumor cell lines, B cells, T cells, and macrophages. And lastly, P53, I'm expecting to be normal to low levels due to its implications within tumor cells, as being mutated within 50 to 60% of all cancers. Being unable to bind to DNA will result in relatively low levels of P53 expression within tumor cell lines. Thank you very much.